Hey, it's Paul. Here we're going to look at some more pathogens. This is actinomyces, so it forms these fluffy little clumps of bacteria. They can look a lot like collections of coccoid bacteria, so you must identify the radiating filaments that branch out from these groups. So you can see them there, they're very fine little filaments. We report these as actinomyces-like organisms. Next up is coccoid bacteria, and so you'll see a bit of this in all specimens. You can see some of it here clinging to the outside of the squamous cell, and sometimes this can be really marked. So notice how dark that top cell is because it has a lot on it. This is Candida, so a large group of cells. You can see the pseudohyphae radiating out from it. We want to make sure that it is parallel-sided and it is segmented. You can see that as I'm focusing up and down, and it has little branching knobs on the side of it. This also has down there a collection of spores, yeast, but we don't report on those. We need to see the branching segmented hyphae. So there's a lot of things that look like this in cervical smears. That's why it's important to see the branching and the segmentation, make sure it's not a hair or a fibre or something like that. You can see there's quite a lot of it collected here. It often collects around cells, and this is a characteristic appearance here that you see, which looks like kebabs, some people think, where the individual hyphae sort of skewer the squamous cells and they look like they've all been run through by a pseudo-hyphae skewer. But if you follow it through, often you have to focus up and down a bit, you can see that it's branching and that it is segmented. And so that can be a bit of a clue to help find it. Finally, here's another quite clean looking specimen. It's got some little bodies in it, I'm pointing out. They're quite tiny, as you can see, compared to the squamous cells around the place. These are Trichomonas vaginalis. Little clusters of them. Sometimes these are quite inflammatory specimens and sometimes not. So these are flagellates, but they lose their flagella during processing. You can see they appear as sort of grey to blue oval shaped bodies with a really indistinct little nucleus that is up one end of the cell, so like an eccentric pale nucleus in the cells. These ones are probably preserved a bit better. You can see the nucleus more clearly in them, arranged along the bottom of that superficial. See the little nuclei and maybe some granules in the cytoplasm. Just low power, hopefully you can see them. Once you see one, you tend to see them everywhere, and they vary a little bit in size, but they're generally quite small. Often it's the inflammatory changes, and I'll talk about them in another clip, like perinuclear haloing and granular debris that really alerts you to these. So notice down the bottom left there's some granular debris, and here we've got some nice trikes with little pale nuclei in them. So this is quite a difficult diagnosis till you get a bit of experience under your belt, but it's quite an important one to make to help control, particularly in settings like this, asymptomatic trach infections. So you see there's quite a few around the place here. So to summarise, actinomyces-like organisms, we see fluffy balls with radiating filaments. They're usually a response to a foreign body like an IUCD, an intrauterine contraceptive device. Coccoid bacteria are reported fairly variably, I'd have to say, between laboratories, and I like to see quite a